So I hear my local clock is chiming for the top of the hour. I'm gonna give it. My phone still says we're 1 minute off, so I'm gonna give it one more minute for folks to get here, and then we'll go ahead and get started. But if you have questions as I'm going through the webinar today, feel free to drop questions either into the Q&A or the chat. Either way, I'll notice and I can respond to any questions that come up through the presentation. Yeah, hopefully, we're all here today to see a VDJ analysis done in SeqGeq. And I should mention, it's a more advanced workflow. So if you're not familiar with SeqGeq, this probably isn't a good place to start. I'm going to skip over some of the basics. We have other webinars that talk about how to use the basic features of SeqGeq, and you can find them recorded on our website if you go to previous webinars, or you can find on our website, Flojo University. And there's a SeqGeq page on Flojo University, and that SeqGeq page has videos that covers all the basics. And we also have documentation. But for today, for the VDJ analysis, I'll probably be starting off on a little bit more of an advanced topic, and then looking deeper at this more advanced type of data analysis. Okay, so my clock says it's 10 o'clock, so let's go ahead and get started. So hello everyone, I'm Dylan Hinson with the Flojo product innovation team, and today we're going to be looking at using the VDJ plugin and doing VDJ analysis in SeqGeq. And so hopefully everyone here is at least a little bit familiar with SeqGeq. SeqGeq is our single-cell RNA sequencing software. It's similar in design to Flojo, our first product, and takes a lot of those single-cell principles, but then lets us apply genomics information to it and to really get a more data-rich environment. So I'm going to be going through these slides very quickly, and then we're going to look at SeqGeq itself and look at the VDJ analysis there. If you have any questions as I'm going along, sometimes I might go a little bit fast, so feel free to stop me, ask any specific questions. I'd be happy to answer. You can put them in the chat or Q&A. And with that, I'm going to go ahead and get started. Okay. So VDJ analysis, VDJ, typically we're getting this VDJ data from either the 10x or Rhapsody pipelines, and VDJ is targeting the T cell receptors or the B cell receptors on single cells. And let's just talk really fast about what the different regions on the VDJ regions are, just to understand maybe some of the terminology and make sure we're all on the same page, and do a little quick refresher on what VDJ is and what recombination is. So VDJ is targeting the BCR or TCR regions on B cells or T cells respectively. And so it's targeting those regions that are regions of interest for recombination, and often are regions of interest for vaccine design or antibody design, or understanding disease pathogens. And so I think we really care about these T cell receptors and these B cell receptors, and so we will do this VDJ analysis to try to find the top clonotypes, the ones that are responding the best to our antigen, and maybe that's like the clonotype we want to clone for or target for a vaccine. So let's just talk about the terminology. It changes between B cells and T cells. So you can see here I have this list of terminology for B cells and T cells. And so on the B cells, on the B cell receptor, which this is kind of a picture of over here, we have the light chain and heavy chain. And so the light chain is this one lower down here, the heavy chain is over here. And the two chains have two different sets of regions, although they're similar and related. So on the light chain, which for a BCR receptor, we'll either call the light chain a kappa or lambda, to just be more specific about what type of light chain it is. So we'll call it kappa chain or lambda chain, you'll see in our analysis. But both those are light chains, and light chains are composed of the variable, joining, and constant region. So the V and J parts plus the constant region, which is really just the boring part that kind of attaches it down here. And so the variable and joining region, though, are of course are closer to our variable region, which is our region of interest. And then the heavy chain is maybe the one we focus more on, which has the variable diversity and joining regions, as well as the constant region, of course. And so it's these three in combination, the variable diversity and joining, where they meet. That's the CDR3 complex. And that's often what we're most interested in, is the combination of these three regions, which the CDR3 complex kind of describes. And so we'll often focus on the CDR3 region in order to classify the different clonotypes. But we can also look at each of the individual variable diversity and joining regions, because they're of course important and play important roles in recombination. So that's for B cells. The only difference for T cells, I mean, first of all, T cells look different. Instead of the kind of Y shape with a B cell receptor, T cells more like only a single side of this Y. But for the T cell, we have an alpha chain and beta chain, and I think they're kind of similar to light chain and heavy chain. The alpha chain lacks that diversity region, similar to a light chain, and then the beta chain has the diversity region present, similar to heavy chain. And these three parts, again, the variable diversity and joining region together, make up our CDR3 region, which we often use again to describe the clonotype. So that was just a really brief primer or review on the different regions of interest in our VDJ analysis. And really what we're trying to do is trying to describe, the combination of the V, D, and J regions that that we are interested in and are responding to our treatment. Okay, so what files do we need for VDJ in FlowJo? So as I mentioned, VDJ analysis will often come from either the Rhapsody or 10x pipeline. And in FlowJo, we support both. And so you're able to load data either from the Rhapsody or 10x pipeline into FlowJo VDJ data, and we can analyze it there with our VDJ plugin. And they basically function the same either way. Today, I actually have some 10x data I got from open source from the NCBI Geo database. But you'll see it's basically the same either way. It's just slightly different file names between the two. So if you're analyzing Rhapsody data, once you come out of the Rhapsody pipeline, you'll get a DBEC or RSEQ gene expression matrix. And so if you see down here, oftentimes it'll look something like this. Their names will either have DBEC or RSEQ. We typically suggest using the DBEC, moles per cell. And you'll need to pair this gene expression matrix with the VDJ per cell annotations file, which is this file down here. And so one thing that's unique about the VDJ analysis is instead of the VDJ information doesn't get written into the gene expression matrix. So we have to kind of pair the two together. So we're pairing the DBEC file with the VDJ per cell file, pairing that information together, so that way we can look at them together. And I think the main reason they don't write the VDJ information into the gene expression matrix is because there are at least two chains or two primary two chains to the clonotype for each cell. And so then from the 10x, it's a kind of a similar thing. And if we have a gene expression matrix, that gene expression matrix typically it's either an H5 or an MTX file. In my example below, I have an MTX file. It can also be a CSV file, but any three of those will work. We can load it into SeqGeq, and then when we run VDJ, we're going to pair the gene expression matrix to the filtered contig annotations file, which often will look like this. And so again, this just came from the 10x pipeline, and usually it'll be named like this. And so that's what we're going to be pairing to our gene expression matrix in order to perform VDJ. Okay, so you can get the VDJ plugin from the FlowJo Exchange plugins. So if you go to our website, flowjo.com, go to the Downloads tab, go to FlowJo Exchange plugins, you can find the VDJ Explorer plugin, and you can download it there. And it's fairly simple to set up. And I'll just show you today how to place the jar file into your plugins folder, and then it should be ready to go. And so when we're running through the VDJ pipeline, when we run through the plugin, it has a few discrete steps. And I'll go through each of those really fast before we take a look at the plugin. But I would say a big power that comes from this plugin isn't just when we're using the plugin itself, but it's also the information we get out of the plugin that we can then use in SeqGeq for our tools that are in SeqGeq. So the first step in the VDJ plugin is this metadata tab. You can see the tabs on the left-hand side kind of indicate the different steps of the plugin. And all this is doing is linking up those two files. As I mentioned, there's the gene expression matrix and the VDJ annotations file, and we got to link them together. And so we use the cell IDs of the two files to match the information. And so you can see here it's trying to match. It has the VDJ information file, and it's matching it to my gene expression matrix. And you can see this is the number, the mapped is the number of rows it's able to match. And so within my VDJ file, in this example, I actually have more rows than I have cells in my population that I'm matching it to. So it only matched this many. And then it also will show us the number of complete clonotypes present, as well as the total clonotypes, which would include the incomplete clonotypes. And so each row represents one chain. And so if we pair the two chains together, we get one complete clonotype. And so that's why this number is approximately half of this number, although there can also be non-unique clonotypes. Some of the clonotypes will be repeated, so it's even less. Okay, so the second tab is our cells tab, and this is where things get more interesting. So when we're using the VDJ plugin, when we get to the cells tab, this is really actually just the information that's contained within that VDJ annotations file. So it's basically, if you opened up the file in Excel, you would see similar information, except Excel is going to really handle it pretty poorly because it's a really big file with a lot of information in it. But you can filter and search this table, and you can sort by, for instance, right now I'm sorting by the unique molecular identifiers and just listing from top down which ones had the most hits. But you can sort and you can search the different variable regions. You can kind of filter and focus on certain regions if you had a region of interest, or maybe a CDR3 region of interest. And you can really get all the information about each of the individual chains in this table here. We can also create gates from this table, but each of these individual rows are representing just a single cell usually. So oftentimes when we gate this, it's not that interesting. But we could filter and gate this table. I think the more interesting table, though, is the next bit of information, which is our clonotypes. And so in the next tab on the SeqGeq plugin, or the VDJ plugin, we're looking at our clonotypes of interest, and we can filter and look at our top clonotypes. And typically when we're doing a VDJ experiment, what we're really interested in is, which are our top clonotypes, which are the clonotypes that are responding the most to whatever we've done to our sample. Usually it's maybe some sort of stimulation or a disease condition. And we're trying to see what our top clonotypes present in the sample are, because maybe those are the cells that are being activated by our antigen. So within this, again, we have this table on the left-hand side here. We can rank it by the number of cells that have each of these clonotypes, and we can kind of find our top clonotypes here. We're able to gate these and bring them back into our SeqGeq workspace as populations. So we're able to subset the populations of cells that contain these clonotypes. I should mention, with VDJ as a whole, it's, there's a lot of different VDJ combinations. There's a lot of clonotypes. It's pretty rare to get multiple cells having the same clonotype. And so even having 5 or 6 cells with one clonotype, that's usually pretty significant for VDJ data. Even sometimes 4 or 3 can be kind of a significant number of cells having the same clonotype. So we're looking at very, very few cells sometimes with this VDJ analysis. Also in this clonotypes tab, we can make all kinds of charts comparing the different regions present in our top clonotype to see if maybe there's some overlap on the variable region, for instance, here, to see what our largest contributors are. We can also take these charts and we can save them to SeqGeq or save them to our computer for future analysis. And then in this last tab, we can compare populations. And so if we already have some gating in SeqGeq, so maybe we've already identified some populations of interest in SeqGeq. For instance, here I've done some clustering already, and I've gotten some clusters on my data, and I want to compare them maybe to see what the most common receptors are within my clusters, or the most common different regions are within my clusters. I can do that and look at, kind of comparing the different regions present in the different clusters. And do all kinds of comparison between populations I've already gated in SeqGeq. Okay. I should mention, this part of it, we make all kinds of graphics. It does get kind of slow the more cells we're trying to look at, because with these populations, as we're adding hundreds of cells, that's a lot of different regions to be trying to display at the same time. And then I think the last and most interesting step really of our VDJ analysis is taking that VDJ information from the plugin and bringing it back into SeqGeq. And then we can do things like differential expression on that clonotype. So here I've done differential expression for my clonotype of interest, and I'm looking at my upregulated in clonotype 1 and my downregulated genes in clonotype 1. And so I have, for instance, this list of genes that are upregulated in what I've called clonotype 1, which is just my top VDJ clonotype, the one I might be interested in. And I should mention the p-values can get pretty small though, because I'm looking at only a few cells, right? Only 17 cells. So my p-value is fairly small, but still we can get some interesting results. And we can kind of look at some of the top genes in this clonotype, most unique to this clonotype. We can also over here, I've done dimensionality reduction, I've done a UMAP, and we can kind of compare where the cells for each of these clonotypes, which I have listed here, are falling on our UMAP, on our dimensionality reduced space. And so that I can compare it to my clustering, and I can also see, okay, for instance, in this example down here, my most expanded clonotype is located for some reason in this region down here, kind of my UMAP. But you'll see most of my other clonotypes, most of my other top clonotypes are kind of clustered here in those top regions. And so I'm wondering, what the difference might be between these two regions. Maybe it's just memory versus naive or something like that, that's separating these different clonotypes. And I can dive deeper into that by doing my typical SeqGeq analysis, looking at some of the changes in gene expression between these two regions, looking at what's upregulated, for instance, and just trying to describe those differences. Okay. So I think that was it for the slides. I'm going to jump now into actually using the SeqGeq plugin and looking more at VDJ. If you have any questions, I've been going kind of fast, feel free to drop them into the chat or the Q&A, and I'd be happy to answer them. Also, if after this, you have any further questions or you want to maybe meet one-on-one and talk about VDJ or your analysis more, feel free to email us at SeqGeq@bd.com. That's SeqGeq@bd.com, and we're always happy to answer questions. I'll put it in the chat real fast. SeqGeq@bd.com. And you'll typically get me answering, so I'd be happy to answer any questions. All right. Okay. So as I mentioned at the very beginning, I've already done some analysis here for VDJ. So I've already kind of set things up. So I've already done some of the basic SeqGeq analysis. So what I've done, the data that we're going to be looking at today, I got from the NCBI Geo database. It's this one over here is the one we're looking at today. And what this data is, it's from Stanford, it's from Gate et al. And what they were looking at was cerebral spinal fluid. So they're taking cerebral spinal fluid from 3 types of patients, from people with Alzheimer's disease, people with multiple sclerosis, and then some healthy controls. And so I've labeled them AD for Alzheimer's disease, HC for healthy control, and MC for multiple sclerosis. And I've loaded some of this data into SeqGeq. So there's a lot of data here. I'm not going to look at all of it today, but I've taken some of these files. For instance, if I go into my Alzheimer's disease folder, there's 4 different patients. I've taken this first one here. I've taken the matrix.mtx file. I've loaded it into SeqGeq, and I've done some pre-processing of the data. So I've done normalization. I've done some gating. I've added a label here just so I know what this is. This is Alzheimer's disease 6. And I've gated the T8 cells. So this is a targeted, they took the cerebrospinal fluid, they sorted for T8 cells. This is targeted, a targeted T8 panel. There should only be T8 cells here, but just in case, I kind of gated to make sure I don't have any dead cells. I did a live dead gate here too, using the mitochondrial genes. I removed anything with a high number of mitochondrial genes because that tends to be the dead cells, and kept my T8 cells. And then as the last step, I did some clustering. So I ran Seurat on this data to get some Seurat clustering. I generated some Seurat clusters, which we can look at later. I've even done a little bit of work to identify some of these clusters. I think this one might be a dead cell cluster, and these two might be some type of memory cell, but I didn't really go into much detail there. But now at least we have a starting place where I've done some analysis, and now it's going to be interesting for me to begin to look at my VDJ analysis and look at my different clonotypes that are present in this data. So I think the key thing here is that we've focused down on a population of interest. In this case, it's my T8 cells. And now that I have this population of interest, I can go further with my VDJ analysis. Again, if you're new to SeqGeq and you maybe want more information on how to do normalization, how to draw these gates and get down to a population of interest, I'd encourage you to go look at our Intro to SeqGeq webinars or our videos on our website that have all the kind of more fundamental steps of SeqGeq, because I'm going to launch off here directly into the VDJ analysis bit. One last thing about the data that I'm looking at today. So I mentioned there's some different conditions. So I've loaded a few samples of each condition. So if I wanted to compare later, I have a healthy control here. I have a multiple sclerosis control here. And so I can look at these different conditions if we wanted to kind of compare the different samples between the different patients. And I could look at the different VDJ populations between each, because each one has within the folder here a contig annotations file. And so again, it's from the 10x pipeline. So they have these contig annotations file. This is the VDJ information that I need to connect my matrix.mtx file to in order to do my VDJ analysis. And so this is going to be great. There's a lot of information here, so I think this is a pretty cool data set to work with. And there's a lot of cool data sets on NCBI Geo. I actually have a B cell data set lying over here too from NCBI Geo. And so if you just search in that database, you can find a lot of good data, or you can generate your own data, of course, with either the Rhapsody or 10x pipelines. Okay, so let's go ahead and get started with VDJ. So in order to set up VDJ, the first thing we have to do is go to the Flojo website, download the vdj.jar file, and then we take that vdj.jar file and we drag it into our applications, and we can put it into our plugins folder. And so we should have a plugins folder. I think it comes with SeqGeq, and we have some initial plugins like the Lex deconcatenate plugin. But if you don't have one, you can always make your own plugins folder and then direct SeqGeq towards it. But I've just drag and dropped my VDJ jar file in here. You can see it. And that's the jar file that I'm going to be using today. The newest version of VDJ is VDJ 4.1. VDJ 4.1 is made to work with SeqGeq 1.8. If you have an older version of SeqGeq, you'll want to use an older version of VDJ. I think VDJ version 3 is for SeqGeq version 1.7. And then VDJ version 2, I think, is for SeqGeq version 1.6. But I'd highly encourage you to upgrade to the newest version of SeqGeq, SeqGeq 1.8, to use the newest version of VDJ, VDJ 4.1. Okay, so with that, we're all set up to use VDJ. So I'm going to go ahead, when I get started using VDJ here, I want to select my population of interest. If we had, sometimes you can run VDJ on a whole PBMC panel. So you'd have multiple populations like T cells, B cells, etc. In this case, it's a targeted T8 panel, so we should only have T8 cells here. But you could run a setback on like all your PBMC cells, or you could focus just on your population of interest. So here I have the T8 selected. I'm going to go up to my plugins and choose VDJ. And what this will do is it will open up a new window where I can select my either my contig annotations file, or if I'm using Rhapsody, it's going to be called my VDJ per cell file. But I'm choosing that CSV file with my VDJ information in it to connect to my gene expression matrix that I already have loaded in SeqGeq. So here I'm in the correct folder. I'm in my 86 folder. And here is my contig annotations file that I need to link to my gene expression matrix. So if I hit open, it's going to read and link the two together. And we can see here, it's able to map, most of the rows in the file to my information here. So the number of rows should be roughly double the number of cells present plus 1. So I think actually the math here, it looks like it's about double the number of, there should be 2 rows per cell. So because there's 2 chains per cell, so it should be right twice, and then there's a header row, so plus 1. But sometimes you'll see it could potentially have more rows, because perhaps when we did the VDJ, when we did our initial sequencing, we had more information, but we discarded some of that information during cleanup. And so that often happens. So we might have extra rows that we're just not going to map. And so the mapped rows are the ones that is able to match to our cell IDs. So it's matching the information in that file to our cell IDs. And once it gets it connected, it should have our complete clonotypes that we can then look at. If we mess up and choose the wrong file, which often can happen because maybe we have many VDJ files and many gene expression matrices that we have to link together, if we choose the wrong one, it's going to be pretty obvious, because it will say mapped and it will say 0, or it will say maybe something like 10. And if we, if most of our cells aren't mapped, it's probably the wrong file that we've chosen. And so then in order to fix that, we could just, remove the file that we've connected to, hit this add metadata button to go choose again and choose the correct file. But in this case, I've of course chosen the correct file, so it's mapped and it's ready to go. And so I can just go ahead and move on to my next tab, which is my cells tab here. Okay, so within this cells tab, this is basically, again, what you get if you just double click on that VDJ annotations file and look at the VDJ information in Excel. But the difference here is we're able to filter it. We're able to kind of search through this a little bit easier. Excel is really slow sometimes. And we're able to, get this information back into SeqGeq. So, so just going to briefly go through some of the columns here. You can see the type. There's TCR and there's BCR. In this case, all of them are going to be TCR though, because I only should have CD8 cells present. Each of these columns I can filter if I right-click on the header. So here I right-clicked on the header. You can see there's only TCR, so there's nothing to filter here. But I can go to my next column, which is my chain. I'm looking at T cells. So there's either the alpha chain or beta chain. If I had a whole PBMC here, we might see alpha chain, beta chain, heavy chain, all the different regions. And so I right click on the header here, you can see there's actually delta and multi that I can look at too. But most of these should either be alpha or beta chain. And so again, that's that chain on the receptor. For B cells, it'll either be heavy or light. Okay, then we have reads. So the reads and the UMIs, so they correspond to each other. The reads are, after our PCR, it's the number of total reads we get for each of these single chains, single RNAs. But then the UMIs correspond to the unique molecular index. And so this is basically the number of original RNA reads that we had that we then amplified with our PCR to the total number of reads. So this is probably the more interesting number to look at. And so the ones with the highest UMIs perhaps were the highest ones were the chains with the highest abundance before we did our PCR. And then you can see listed here, of course, the variable diversity joining and constant regions. Our VDJ variable diversity joining. We can focus and filter these regions. And so if I just right click here, for instance, I can focus on some of the specific variable regions. If I had one that I was really interested in, I could remove some of these other, I can say none, and then I can just choose one, hit apply. And now we focus down just this one variable region. And you can see it changes the whole table. I can look at 2 or 3 of them if I wanted to. And so let me go ahead and say reset to all. We have a diversity region here, which sometimes is the region we're most interested in out of these 3. We also have joining, and then constant, which usually is the most boring, and then the CDR3 region, which again, we can right click and search for specific CDR3 regions that we might be interested in. And this is often the region that people will use to identify the different clonotypes. And then, of course, to the right of the CDR3 region, we have the entire nucleotide sequence of the chain. And then sometimes the annotation will have additional information about what the platform, like the Rhapsody platform, for instance, thinks our cell type is. And so sometimes the platform, when it exports the data, will make guesses for us about what the type of cell it is. But for this data, there's not that information isn't there. Okay, so I guess just to show one other feature, we can select rows and we can hit gate from selection. And what that will do is over here in my SeqGeq workspace, I now have this gate, which is a single cell, right? Because we chose one chain. So one chain usually means one cell. And so we've gated a single cell here now in SeqGeq that had this chain present on it. And hypothetically, we could look at that single cell more closely, or we could select multiple cells, maybe that all have the same variable region. And we could make a gate from multiple cells, for instance. Let's actually look at so none. I'm just going to choose kind of at random one of these variable regions. And so we can make a gate from the selection here. And then we have, I guess there's 41 cells in that list that have that same variable region. And so now we can focus on that one variable region if you wanted to. We have a population here for the cells from that variable region. So one thing to note about when you're using this gate from selection feature on the cells tab is it comes up with a random name for the population that it creates in SeqGeq. And that's because it doesn't really know why you wanted to select these cells or these chains to put them together. But I know in this case that I chose, I filtered for the TRAV12-1 variable region. So I might just call this TRAV12-1, just rename it in SeqGeq so that way I know why I chose this and what might be interesting about it. I'm gonna go ahead and delete this. And so that's one of the key things about this VDJ plugin is that we're able to take that VDJ information and use it to make gates on our sample back in SeqGeq, and then we can do later analysis with those populations that we're making in SeqGeq. Just really fast, since I've gated one variable region, I might as well gate another just so we can compare them later. I'm going to randomly just choose TRAV2 since it's next in chronological order. And so then maybe later we can take a look at comparing these two different variable regions, which are probably pretty similar. Okay, so moving on. So, I can do a lot of filtering and looking at different populations here in this cells tab, but I think the really cool tab is this clonotypes tab, because this is where we get our top clonotypes. And this is probably what we're often the most interested in is, which clonotypes are really being are really present or being expanded in our data. So you can see here there's a list of our top clonotypes. We've sorted, if I double click on these headers, I can sort by the headers, but I want to sort by the cells just to have my most abundant clonotype at the top, and then the list descends down. And you can see it lists what type it is. So these are all going to be TCR because I'm looking at CD8 cells. It has a list of what chains are present. Again, every one of these should have an alpha and beta chain. It's the paired combination that makes the clonotype. And then the CDR3 regions that are present on these clonotypes as well. And then over here on the right, we can see there's a pretty boring pie chart right now of the variable regions. You can see the x-axis here is what it's using as the pie, the statistic for the pie chart. And you can see I can manipulate these pie charts though. So let me go ahead and add more information to that pie chart, and it's going to get more interesting if I select multiple of these. And the way I'm going to select multiple is I'm just going to hold down the shift key on my keyboard, click on everything that has at least 4 cells. So here's 3, so I'm choosing everything 4 and up. And I can look at them all on this pie chart now together. And so we can see some of the variable regions that are maybe shared between these. What are the most common variable regions? In this case, there's a lot of unique variable regions. It looks like these two are maybe the most abundant, but most of them are pretty well shared. And then we can also switch, though. I can switch my x-axis to look at some of these other regions, like the diversity region. Yeah, so it's either none or, most of them are, half of them don't have a diversity region because of the beta chain. But anyways, we can look at the different diversity regions, CDR3 regions, and begin to identify maybe regions of interest, some of our most common ones. And so let's just focus maybe a little bit more narrowly on the top 5 here. I can go ahead and I can save this image as a PNG on my desktop, sure. And then if I wanted to, I could always use this image later in a presentation of some sort. I can also click this To Layout button. And what that's going to do is it's going to take the image and put it in my SeqGeq layout, because it might be useful to have it there with the rest of my SeqGeq analysis. I can mess with the plot type, change it from pie to bar, do things like that. And so, yeah, those are just some of the different options there. But I think the real more interesting thing about this clonotypes tab that I should probably talk about before I run out of time is the ability to gate these clonotypes. So for instance, if I'm interested in any clonotype that has at least, let's say at least 4 cells present, I can select these clonotypes here. Again, I just shift click to select multiple, and then I can right click on the clonotype. I accidentally unhighlighted everything, that's fine. So I'm right clicking on this top clonotype here, and we can see I have 2 options. I can create a gate from clonotype or a gate from clonotypes, plural. And if I choose this gate from clonotype option, what it's going to do is it's going to make a gate of this clonotype here in SeqGeq, and it's going to list the 2 CDR3 regions that are combined to make that clonotype. And so now you can see I have 17 cells in this clonotype. there are now 17 cells for that clonotype in SeqGeq. So that's my clonotype there in SeqGeq, it's those cells. And now later I can look at that top clonotype and I can begin to do all kinds of analysis with that top clonotype. I can also, highlight some of these other clonotypes. So I'm going to select all the rest of them here. Then I'm going to right click, whoops, and say gate clonotype. If I choose gate clonotypes, instead of making individual gates for each one, it will combine them all together in one population. But in this case, I want individual gates. That's usually more interesting. I can always combine them later in SeqGeq. Gate clonotype. And so now you can see I have all my clonotypes listed here in SeqGeq. It becomes a lot. It can be a lot of data really fast, but that's probably fine. The more data, the better. And so now I have my clonotypes gated in SeqGeq, and so now I can do differential expression and gene analysis. I can look at them on clusters. I can look at them in a dimensionally reduced space. I can do all kinds of things to try and identify what makes those clonotypes unique. Let's go ahead though and let's finish up here in the VDJ plugin before maybe we look more at SeqGeq and how we can identify and analyze things in SeqGeq. So just to finish up my VDJ plugin, I'm going to go to the last tab here, which is populations. And so this tab can get a little bit messy pretty fast. So I'd be careful when using it. But what this can do is it can show us our variable diversity joining regions within a given population. So for instance, if I've already done some analysis in SeqGeq, for instance, I've already done clustering here in SeqGeq. And if I want to maybe take a look at one of those clusters and see, what cells are kind of present in that cluster, what regions are present in that cluster, I can take a closer look at it with this populations tab here in VDJ. And so you can see all my populations that are here in SeqGeq are listed in this populations tab for the sample that I'm running on. And that can be a lot because I have all these populations from my VDJ analysis, but I also have all my clusters. And so what I'm going to want to do is I'm going to want to just choose one or two of these populations maybe to take a look at and begin to compare. So in order to actually edit and select these populations, I'm going to have to flip this edit populations tab button here. And that lets me go ahead and choose some of these populations to take a look at. And so I'm going to go down here to my clusters, my SeqGeq clustering that I've already done before today, and take a look at one of these clusters. So I'm going to choose cluster 4, which are my, hopefully some memory cells based on my previous analysis. And let's just take a look at what's in that cluster 4. So now I've chosen it. I can choose a color for it. The blue seems fine to me. And then I'm going to go ahead and say finish editing populations. And it made it over here, this bar chart of my different variable regions, because again, you can see where it says x-axis here. I have variables selected. I could always switch it though to one of these other regions. And then of course, if I wanted to compare two clusters, I could hit edit populations again. I could choose a second cluster to compare to. So let's choose cluster 3. It's going to be green, and then I'm going to hit finish editing. And so now you can see it's comparing these two clusters to each other. And so I can see maybe what variable regions are present in one, but not the other, or what are the most common with the two of them. And so I can make all kinds of, figures here, bar charts comparing the two. Again, I can save it to my layout and just kind of put it here in a new layout. I can also save the image, of course, to my desktop and have it here. Oh, it's the same name as my previous image, so probably good to rename those. So yeah, so that's it's a quick run through the VDJ plugin and kind of some of the features that it has within the plugin itself. But the cool part of VDJ is it's not finished once I'm done with the plugin. So I'm going to go ahead and close the plugin browser unless anybody has any questions about the plugin. If there are questions, though, I can always come back and look at the same information again. Just by rerunning the plugin, it will open the same browser. So I can close it for now and not worry. I can go back and get that same data later. Once I'm done too, I'm going to go ahead and delete this VDJ population just because it might rerun and reopen that window, which can get kind of annoying sometimes. Okay. So let's kind of take a look at some of our VDJ data here in SeqGeq now with our remaining time. So the way we can take a look at it here in SeqGeq, I've already done some pre-analysis on my data. So I'm going to open up my layout editor here. I'm going to take a look at my, my T8 cells. I've done Seurat, which means I've done a UMAP as well on these cells. So UMAP X, UMAP Y. I'm going to copy this UMAP to my layout. This is of all my T8 cells. And then I can, highlight some of these specific VDJ populations on this UMAP. So I can drag and drop just one of these onto the UMAP. The colors are kind of awful right now. I might switch this to gray. In fact, I kind of like to make it a contour plot. So if I just click twice on this graph window, it opens up this graph definition window. I can change from dot plot to contour plot, and then it makes it easier to see where my VDJ populations are in this UMAP. Let me just grab some of my other top clonotypes, drag them in, and we can see, where some of these cells are kind of clustered on this UMAP. And some of them are kind of clustered together, with each other. Others are a little bit more spread out. Again, we're looking at pretty sparse data, but we can maybe begin to identify where those clusters fall, especially if we relate them to some of our populations of interest, our clusters of interest. So let me go ahead and I just cloned this same graphic. I'm going to remove my VDJ information. I'm going to go ahead and add my Seurat clustering information by dragging and dropping to overlay it. And we can kind of see, okay, so here's my Seurat clusters, and we can look at maybe how they compare. So for instance, most of my major clonotype is down here in this region, which actually is kind of split, it looks like, between a few of my Seurat clusters, my dead cells, unfortunately, but also looks like the orange and the blue, which are 1 and 2. And so anyways, I can begin to try and identify, and then like up here we have these orange cells, maybe number 3, which correspond to perhaps this clonotype. So I can begin to make those comparisons and try and identify what clonotypes are present in which clusters. Another comparison I can make within SeqGeq when I'm looking at my VDJ data is I can do differential expression analysis, which is going to be pretty interesting. So let me just take my top clonotype. To do this, I'm going to want to go to my T8 cells again, though, and compare all of them. So I'm going to go do differential expression analysis, which means I'm going to want to switch to gene view. On my y-axis, I'll want to put my population of interest, which in this case is going to be my top clonotype, which their names are really long, but I can see this one is in fact my top clonotype. And then on my x-axis, I'll want to maybe focus on my T8 cells. And you can see already some of the genes that are present in my clonotype that maybe aren't present as much in the rest of my cells. But if I press the volcano plot up and down arrow, I can do differential expression analysis and get this plot over here. And what we can see is that the Q value, the significance is very small for the genes that are being differentially expressed. And that's because I'm only looking at 17 cells. So it's hard to get a high Q value when we only have, or a low Q value when we only have 17 cells. And I've actually already done a step to try to make it easier for me to look at lower Q values. For me to look at higher Q values, less significant Q values. And so what I've done is in the heart icon of SeqGeq, if you go to the heart icon in the top right-hand corner, you can go to the graphs preference in the middle, and you can choose this p-value threshold and a p-value adjustment method. And so typically, I think by default, we have either FDR or Bonferroni selected for our p-value adjustment to get that Q value. But I've turned off my p-value adjustments. That way I can look at less significant data because we have so few cells that it's just impossible to get a high significance for my up or down regulated genes. You can also alter the p-value threshold that SeqGeq suggests, but if I hit OK here, we'll see that's how I got this p-value to be visualized. And if I customize axis, I can maybe even zoom in a bit more on where all my data is, since they do have such big p-values, low significance. And so I can go ahead now, hit display, draw large dots just to kind of maybe see it a little bit easier. And there are some that are above a p-value of 0.05. And so that's pretty significant. So I can go ahead maybe and gate those, and just say down in my, I'm gonna call it clonotype 1, and then I'm gonna call this up in clonotype 1. So then we can get some genes that are up or down regulated within clonotype, what I've called clonotype 1, which is this my top clonotype here. And we can see those genes. I have a lot of gene sets already loaded since I've done my Seurat analysis, but we can see those genes listed here in my list of gene sets. I can double click on my differentially expressed genes in clonotype 1. And now I can look at some of the genes that are present in a higher frequency in clonotype 1 than the rest of the cells. So they might be interesting genes to take a look at if I'm trying to identify what makes this clonotype unique and to identify what this clonotype is. And so, just glancing at it, I'm not recognizing necessarily any of these genes, but we can then do further enrichment and analysis to try and identify what makes this clonotype unique. So I've gone through kind of a lot, and thanks for sticking with me through this. Again, if you have questions, feel free to drop it into the chat or Q&A. So far, I don't see any, so I'm just going to keep going, but let me know. We only have a few minutes left, but one thing I wanted to show is that we can run Seurat, or not run VDJ on multiple populations, multiple different files. So I've been looking at that Alzheimer's disease patient 6 this whole time and just focusing on that one sample and the top clonotypes within that one sample. But I could go a step further and I could look at my healthy control now and maybe compare if there's any clonotypes that are shared between the two and maybe see which ones are different, because I probably care more about the ones that are different between the two than the ones that are shared. So let me go ahead and run VDJ again on this healthy control. And we can use some of the information that we got from looking at the Alzheimer's disease patient to maybe identify populations of interest in the healthy control. Okay, so I'm running VDJ again. I need to go to my data, go to my healthy controls, healthy control number 9, and choose that contig annotations file. Okay. You can see it's mapped. I could filter and search here. I could, if I had regions I was interested in, which I may have identified in the last, when I was performing VDJ on the last file. But I'm just going to jump straight to my top clonotypes, see what they are, and see if there's any overlap between my top clonotypes from my first sample to this one. And so, oops, I'm just going to go ahead and start by gating them. And then we can look at them in SeqGeq here, just because if they're both in SeqGeq, maybe it's a little easier or harder. I mean, the names are so long, it's sometimes hard. So look at them both. But just looking here, my top clonotype from the first one is not present in the second. I guess I could go back here, search by CDR3 region in the cells tab to see if that CDR3 region is even present. The CA, A, S, A, and T. No. I could look through some of the top clonotypes I have, though. Nope. CAG, P, GV, nope. And yeah, and there are so many clonotypes in general that, typically there's not going to be a high amount of overlap between two samples. CAVR, DL, GV, slightly different. All right, so different clonotypes, but we can begin to address some of the properties of these clonotypes too. Again, we can, when we're looking at the clonotypes here, we can see what variable regions are present, maybe compare the different variable regions to see what the different properties of the clonotypes are. I think I have that image from the last one, so let me go ahead and make a new image in my desktop. Let me just call this is healthy control 9. And maybe we can compare the variable regions between the two. So healthy control 9. Whoops, I made it of the CDR3 last time though. Yep. So we can compare the CDR3 regions. Whoops, it did not replace it, even though I asked it to. It may not have permission. Okay, so that should have made it. Let me see here. CDR3. So yeah, now we can compare the CDR3 regions that I've selected between the two. Again, I think we already know actually that they're going to be different because I was looking at them in SeqGeq and they were different. Yeah, we can see maybe some of the CDR3 regions actually. Yeah, some of these looks almost the same. There's some CAS regions here. Yeah. And so I guess that's just one way we can compare these different files, look at the VDJ regions that are present, and do further analysis. Okay, so we're getting close to the end of time. I want to make sure I leave a couple minutes for questions, if anyone has any questions. If not, thank you for joining me today. Always feel free to reach out to us at SeqGeek@bd.com if you have any other questions, or if you want to schedule a screen share for us to look at maybe your data together, we'd always be happy to do that. So yeah, thanks everyone for joining me today, and hopefully now you're ready to jump into your VDJ analysis. Okay, I'm not seeing any questions. So hopefully hopefully that means everyone's on the first steps to making a great VDJ analysis. I'll give it one more minute. If you have any questions, quickly type it out, and then I'll go ahead and close the webinar. Alrighty. Well, thanks everyone for joining today. Yeah, please let us know, at SeqGeq if there are any further questions. Yeah, hopefully you're making a great VDJ analysis. All right, have a good one. Bye.